Good afternoon, everyone. It's Dr. Gershon with another BetterHealth webinar on healthcare innovation and technology. I am absolutely thrilled to be here with Samantha Strasser, who is the Chief Science Officer for Pepper Bio, and she's the co-founder alongside her partner, John Hugh, who unfortunately can't be with us today. But it is a real thrill to have Samantha here today. Endeavor is a strong supporter of the work that Pepper Bio is doing, and this is an opportunity for the community at large who's interested in healthcare innovation as part of the Endeavor ecosystem to understand more about the work that they're doing, because we firmly believe over the next many years it will have a strong impact on patient care and patient outcomes. With that, I want to just first of all thank everyone once again. We've had tremendous support in terms of interacting with amazing speakers, incredible companies who are bringing a vision of entrepreneurship and verve and excitement to the healthcare and life sciences space. We do believe that we're in a growth cycle, which is secular over the next 10 to 14 years in healthcare, and particularly in digital health. And so as part of this, Endeavor has a venture studio, which is sponsoring today's event, which looks at helping pre-seed, seed, and Series A companies grow over a 4-year period. And we have a Series B venture fund called Endeavor 2, which then extends out that relationship. We believe that the webinars are a source that allows different companies to interact with other great entrepreneurs, other industry leaders, and investors, as well as scientists, doctors, and people who are surrounding the healthcare profession. And we believe that this open source ecosystem is something that is going to contribute to better healthcare. With that being said, I want to acknowledge the incredible work of Pepper Bio. They have really gained a lot of traction for a young company. And I think I want to begin by introducing Dr. Strasser to you, because she is really an incredible scientist and entrepreneur, and she will tell you her journey, which is really outstanding, and also will tell you about the innovation that she actually has created. She will tell you how that innovation is going to impact you as a participant, as a potential patient, unfortunately, in the future. And she will describe to you a little bit about what her needs are for the company over the next several years. Because in this collaborative environment, it is our firm belief that the only way to solve these complex problems is to come together. So thank you once again, everybody, for dialing in. I see already we have a number of people, and I just want to quickly thank them. Aurel, thank you. Nikhil, thank you for dialing in. Chad from Dallas. Jenny, of course, from New York. Thank you all for dialing in today. And I would encourage people who have logged on to please let us know where they're dialing in from, ask questions, and if you want to include your email, we can send you an email response to any question that we don't answer today. So with that being said, I want to introduce you all to Samantha Strasser, who is the Chief Science Officer for Pepper Bio and the co-founder of Pepper Bio. Welcome, Samantha. It's a pleasure to have you here today.
Thank you, Dr. Gershon. Great to be here. And thanks to everyone for joining, emphasizing the goal from these events today and the like. It's really people at the end of the day that are what bring together new solutions and innovative technologies. So thrilled to be sharing this with such a great community and look forward to connecting with folks in the future. So I guess I'll start just by a little bit of background about me and Pepper itself, so you can have some context of how things got started and why I personally am here today. So just taking a bit of a step back for me by way of background and interest, I've always been really thrilled about simply the data and technology that we now have to tackle really complex diseases. Biology is in a phenomenal spot of having a wealth of information to draw from and computing power to really go after teasing out new insights from that. And so for me, my journey started essentially as early as my bachelor's degree at Northwestern University. So I knew early on that I wanted to delve into the healthcare space to really make a big impact for patients. Hadn't known quite what that meant over 15 years ago now, but knew that I wanted to build out my career and really looking for the technical skill set to delve into these technologies and data and the ability to bring the technologies I would work on to patients at the end of the day. And so that early vision is actually what first brought my co-founder and I to want to start a company together. We met back at Northwestern University during our undergraduate degrees in biomedical engineering. Myself, I paired that with applied mathematics, and John had paired his degree with economics as well. And so we had really a synergistic goal, but two sides to that coin of really being able to build out skill sets to bring this future venture forward. And so for me, following my time at Northwestern, built out on the technical side, earning a master's at the University of Cambridge in the UK through the Churchill Scholarship, then came back to the US for my PhD. And it was while starting my PhD at MIT that I really had two fundamental experiences that shaped to where Pepper is today. So first experience that really shaped was that on the personal side. So I learned very early on in graduate school that my father had frontal temporal dementia. So this is a disease that, like Alzheimer's and other neurodegenerative diseases, has no options for patients, and is a scenario where you go to the doctor, you're diagnosed, and you're sent back home. And the reality of what that meant for my father and for our family of having no way to curtail the disease and witnessing that experience really shaped what I wanted to do with my career. I'd always loved the technical side, been excited about the science and the technology, but really honed that at the end of the day, that what I worked on, the science I studied, the technologies that I would found, needed to reach a patient to change someone else's future and someone else's family's future, so they don't have that experience of not having options for their disease. Unfortunately, my co-founders had similar experiences as well in neurodegenerative disease space. His grandmother was diagnosed with Alzheimer's disease, and similar experience of witnessing the decline of a family member that we both unfortunately share, but has been for us really a core that drives forward what we're doing in our careers and also here at Pepper. So I'll build out further on the technology on Pepper's side as well. But personally, that's really what brought each of us to founding this company. The second facet for me in graduate school that really shaped to where Pepper is today is on the technical side. So it was the work that I was doing for my PhD. I was earning my degree in electrical engineering and computer science at MIT while working with Doug Laufenberger in biological engineering and co-advised by faculty member at Harvard Medical School, Kevin Haggis. And with the experiences with my father, really delved into a lot of questions around why are we failing patients today? Right? Fundamentally, why are these diseases still without treatment? And what in the technological side and the insight side are we missing? And so delved into seeing genomics, the phenomenal technologies, the foundation to a lot of our discovery today. But it's one piece of this really complex puzzle. It's the instruction set to be kind of the set of what can happen in disease. But fundamentally, where we need to go is looking at what is the carrying out of these instructions to truly get at understanding complex disease. And so my thesis work for my PhD was focused around how do we better capture disease-relevant information beyond the instruction set to truly tease out functional changes in disease to best know how to treat that, and then how to understand potential drugs that will result in a successful treatment for patients. And so going after that in my PhD, specifically looking at data types beyond genomics that are more functional in their focus, specifically looking at information on proteins, so molecular actors and modified proteins, Really big word on is phosphoproteomics for those who are familiar, but delving into what happens when you add molecular switches to proteins. What is the change of their function and what they're doing in disease to best know how to have a really correct treatment to resolve that disease. So early work in my PhD was successful in identifying novel treatments for inflammatory disease, IBD, as well as novel insights for difficult to treat cancers, mutant KRAS cancers in oncology. And that was the foundation to seeing there was a technology that was bringing new insights. My co-founder, John, had been working in pharma at the time and saw really the need that the industry had to bring these technologies to their own questions. And so that really synergy of both the technological advance and the need in the market is what brought us to founding Pepper to build upon this technology and expand it to bringing transomics to life. And so that's what here at Pepper, our foundation to what we're doing is that fundamentally is bringing an end to untreatable diseases for our mission. And we're thrilled to be sharing that with folks such as yourself today. I'll pause there for a minute in case Dr. Gershon's any questions you have. It's a lot at once to start with, but thrilled to be here.
Samantha, thank you so much. It was a fantastic overview, and your background is incredible. Both your personal journey as well as your academic career really, I think, is a testament, to authentic, mission-driven work. And that's what we're all about in building out this ecosystem of collaboration, bringing people together who share that ethos. So, I know this is the first time and certainly not the last that you'll be on the webinar. We also have podcasts that we're initiating in the fall. And I know I spoke to your co-founder, John, and you about that and follow up, because I'm sure that the way that your company is moving in terms of the traction that you're getting is something that we're going to be wanting to talk about a little bit today, but also in follow-up of the months to come. I think that what's exciting here is that, bringing together scientific skill sets, business skill sets, the industry experience, that John brings, coupled with, the powerful ecosystem that you've come from both in the States and out of the States, I think, speaks to how do we solve these problems together. It requires people from, committed visions, but different backgrounds to bring skill sets together. And that I highlight that because, intuitively, the focus of Endeavor is to find companies to work with like yours. And there is, I think, a connectedness in terms of the, shared way that we begin this process. This is not a short-term, event to solve these kind of problems. It takes years. I am happy to say that the technology that I see in companies that have been started just a few years ago is already, groundbreaking. You just have to look at one of the companies within the drug discovery, drug development space, which is also crossing over to what we're going to be talking about now, which is in silico, and see that the work that they're doing to use AI and advanced technologies has allowed them to use AI to target new, drug opportunities, and then to validate that, and then to move it, into not just preclinical, but into the clinical realm, and then commercialize all within 30 months. So that's
we can do today as an industry.
So that's a segue into, what is Pepper all about? And I'll just give you my 1-minute overview, but then you can, dig deep. So I am excited about working with Pepper because they first of all see the big picture, and then they have small step operational solutions to that big picture. So the big picture is, how do we identify, the proper targets to go after to control diseases? That's a huge issue. The second is, how do we use these identification techniques? And they may be kind of multifactorial. One of the areas that Pepper is focusing in on is biomarkers. So how do we use biomarkers in a very goal-oriented, action-driven way? So that's kind of step number one that I'd love you to talk about. And then step number two is, once we get this information, how do we use your novel approaches to looking at, this intersection between protein folding and kind of molecular biology, molecular chemistry. I mean, there's many different scientific integrations here, which are fairly complex, and we don't have all that time today to go over. But I think high-level overview of the importance of proteins is well understood, but then this other piece that you bring into it, to control proteins, I think is extremely exciting. And then, how do we use that information of identification and what I would call moldability, or the ability to shape this process? How do we use it then to find therapeutic modalities? So this is a lot of information, well, within your super focused area of expertise, beyond most of us in terms of understanding the real deep science. But if you could give us an overview, because I consider this as groundbreaking, as in silico.
Thank you. And I mean, it's, echoing this phenomenal time we have of what we can understand in the industry today. By way of context, we're actually a phenomenal complement to a technology like in silico, as we're able to actually, as you said, understand the disease and the target and how to identify that disease through, say, a biomarker. And then the other side, so after there'd be an acid or molecule, what we do then is come in and say, what is this drug doing? And we actually look at a global signaling scale that's comprehensive to ensure we have the full scope of the drug's impact on a potential disease. And that's actually what I'll be talking about today as well, is sharing a case study that actually looks at how we can help to select a successful drug from an unsuccessful drug, and to very early on in the drug discovery process to provide that answer of, what is my drug doing, and do I want to move forward with this drug?
Fantastic. I mean, that's just such a big piece of the equation. And I will tell you that, so approximately 60% of the costs associated with drug commercialization are preclinical.
Yeah.
And so, looking at this space carefully to be able to create more efficient models brings drug to market faster, which means faster treatment algorithms that are novel.
Mm-hmm.
As a former FDA attorney, I can tell you that the regulatory environment is very much putting the wind at the back of companies to do these things. And at the same time, there's another piece which I will just put in, as a important but side piece, not directly pertaining to your company, but the FDA is a very supportive mechanism now, which includes the recognition that not only finding the right drug, but doing the right type of study is critical.
Yes.
So they have a new initiative, which we are completely supportive of, that mandates diversity, equity, and inclusion plans in all clinical trials. And so as part of this ecosystem, I think there is a recognition of really the importance of getting accurate data,
Mm-hmm.
which not only speeds the process forward, but is also addressing issues of access. And I think, while this isn't the core focus of your company, I think access is embedded in what you're doing, because you are bringing things in a more focused, accurate way. It's speeding the initiative. You're making quicker decisions. Extending that out, is a lot of very interesting things happening out of government, like, for example, the new initiative in Washington, which is financing internet across the country.
Mm-hmm.
Again, these are all access points. I don't want to take us too far off the main point, but I guess my overall point is that there's a lot of different forces coming at healthcare in a very, very positive way. So big picture, but then turn it back to you for really dialing down into the science, and you can, start wherever you like. There's so much to talk about.
Yeah, absolutely. And I guess I'll build on that briefly, and then we can transition to the slides to talk more about the drug selection story as well. But just the note I'll add is, one of the benefits to our approach is that we are selecting the patient population as well as something that we can bring new color and understanding to to ensure that it's reaching the right population of patients that can receive the treatment and they will respond to it and it will be successful in the long term. I'll touch a little bit on that at the end of my slides as well, but that ability to appropriately select patients from to receive the treatment is also a huge factor to ensure that we're reaching the collection of people in need.
Great. I'll put the slides up if you like.
Perfect. Excellent timing. Thank you. So delving into an example of a case study again of how we've leveraged transomics to identify successful drugs. I'll start with a big picture really on kind of bringing some numbers to the problems that we're facing today. And specifically, this is within a EGFR mutant non-small cell lung cancer. So just illustrating the burden that even on the clinical side, let alone the preclinical numbers that you had mentioned, Dr. Gershon, but just how much it costs essentially when there are failed drugs. And so in this case, an EGFR mutant non-small cell lung cancer, it was roughly 2,000 patients that received a drug that was at the end of the day not approved. So did not have an ability to have their disease resolved effectively. Furthermore, it was an expense of over $400 million. And if we sum up the total number of years over these 3 failed programs, that cumulative time sums up to over 40 years. So again, really there's the human expense, the cost, and the time expense of really what happens when in the clinic, a drug is brought forward that at the end of the day was not successful, in part because not enough was known prior to entering the clinic to understand whether or not it would be an approved drug. And so Pepper's technology, really rounds out one of the 2 areas of gaps that we have today, which is that fundamentally these trials often fail because of a misunderstanding of the disease. So not truly having a comprehensive understanding of what sustains disease. And the second category being a lack of understanding of the full impact of a drug on the patient. And so really both sides of this coin on the fundamental level are what really, again, not having enough information on this front can lead to a failure in the clinic. And so building on why does this happen? And so if we think about complex diseases, fundamentally, there's a need for more information, but also the right information and data to draw from. This is something, building on the part of the theme for today about the importance of AI. Another huge facet to it is what data is being drawn from, because it's not just about having more data to run in through an analysis, but also having the right data to start from. And so at Pepper, we kind of categorize the type of information we bring in, the value we bring in 3 fundamental pillars. So one is we bring information and data that's more functional. So we look beyond static or expression information to have truly an understanding of what's happening in a disease or when a drug is applied, to have a functional understanding, to have the right target to go after and the correct characterization of a drug to make an informed decision. Second facet is we bring a global understanding to everything that we do. So we bring large-scale measurements that are not pre-selected, they're not a panel, they're not partial of a favorite pathway. In contrast, they're the whole scope of the entire proteome or phosphoproteome, as I'll talk about later on. And then lastly, what we bring is value on information that's not just correlative, but drives towards a more causal understanding of how different data types interact. As I'll share on upcoming slides, transomics fundamentally integrates across multiple different data layers to truly understand biology at its fundamental level, to answer these questions around what happens in a disease and what happens when a drug is applied. And so just to round out a little bit on the success that this has brought to date and the interest that the industry has shown in this, Pepper's traction is seen dramatically through partnerships that we've had, both with pharma as well as academic labs as well, so Stanford University, totaling about, for pharma partnerships, $1 million in revenue to date. And it's just a couple of key highlights. This is across multiple different therapeutic areas. So in this case, oncology, inflammatory, and neurodegenerative. And notably, we've had the excitement around working with a top 5 pharma partner, as well as other commercial and private companies. And notably, a range of different goals that these are going after. I'll highlight in the last column there. So everything from early on in target ID to understanding drug mechanism as well. So just rounding out a little bit more on the team. So you have myself, as I introduced at the beginning, my co-founder, John, there in the center, kind of building on his background as well. So having both operated and scaled up and grown companies through his time as a consultant and in venture capital, as well as working in pharma, and then most recently earning his MBA at Harvard Business School. We're also joined by, on the business development side, Caitlin Brown. So for those in the community that are interested in connecting on the BD side in pharma, you'll likely connect with Caitlin at some point as well. Phenomenal scientist and turned banker who's now working with Pepper to build out our partnerships. And also supporting our team, we also have a phenomenal advisory board, so notably my advisor from MIT, Doug Laufenberger, as well as Dean Felcher over at Stanford, who is our current partner in one of our oncology programs for Pepper, but also a range of folks on the industry side as well, who have experience, again, really understanding what pharma is looking for and how to best connect with the key needs of the industry. So what is transomics? I've talked about a little bit of this going through, but fundamentally this diagram is how we like to show what value transomics brings above and beyond that of looking at a traditional R&D program. So I'll highlight first on the left a traditional perspective where you'll have, say, a large-scale genomics measurement. So again, all these white points representing data that's collected within genomics. So again, a full global perspective, but oftentimes going up the stack to other data types, say transcriptomics and proteomics especially, there's a narrowing of what information is looked at that's more limited. And also we'll notice here, it's layers of information that are of expression. So more static information of how much is there of a protein or a transcript, but doesn't answer the question of what is that protein doing. And so Pepper's approach, I'll note first, we bring in a top layer of phosphoproteomics. So that's that label there up at the top, which brings in functional understanding. And so this information, it brings in context of modified proteins to understand what happens when a protein is modified, what is its function, what is it doing in the disease. A second facet is that we bring in information across all these data layers that is more global. So that's represented here by showing we have those points throughout the entire set of information from genomics, transcriptomics, proteomics through phosphoproteomics. And then lastly, we bring more causal information. So it's highlighted there on the right, but we have a linking of information by these green arrows as well as the blue that interrelates the information of what's actually happening and how they are interlinked between these data layers to truly get at understanding biology as it actually happens. And so to round out a bit more on this in terms of the contrast with multiomics, if we think about how often a multiomics approach can be analyzed, is 3 data layers, in this case representing genomics, transcriptomics, and proteomics, may be collected. Results are looked at independently in terms of what is significant and relevant. And then an overlap is often seen of, okay, where are they consistent? Now, this misses fundamental information because, one, it's not capturing how biology actually functions. You don't have a functional context in this mix. Second is the understanding and looking at overlap is not necessarily how biology works. You can have an increase in a transcript, and depending on feedback mechanisms, this may be occurring as a result of the protein initially decreasing. So you can have a change in cycle here where an overlap may not be representing what's truly relevant to the disease. Now, in transomics, we look at these data types in a singular analysis and notably bring on that green top layer representing phosphoproteomics in this mix. And so from that, we narrow down to a really selective set of significant pathways and activated enzymes and proteins that are actionable and truly representative of the biology at hand. So bringing in this global, functional, and causal perspective can truly get at understanding the fundamentals of a disease or understanding a drug. And so we'll delve in next to applying this technology to answer the question around what drug would be successful for in this case, EGFR mutant non-small cell lung cancer. And so the challenge that we've often seen historically, as highlighted at the beginning, is late-stage clinical trial failures are very costly, both on the patient time and cost side. And oftentimes, again, these happen because there's a lack of understanding of safety and or efficacy. And so what we did at Pepper is we used transomics to really delve in and identify differences between approved and unapproved drugs that all had the same mechanism. And with this, we were able to see dramatic differences between these drugs, notably from data that was actually a preclinical type study. So looking at in vitro data and being able to interpret this is a huge advantage, being able to make informed decisions early within drug discovery. So highlighting a little bit more on the specifics, compounds that we studied for this case study. So again, 4 very similar drugs with a range of different clinical outcomes historically within clinical trials. So the 3 at the top had not been approved, so EGFR inhibitors, highlighting the stage they were discontinued, and also the efficacy and safety readouts that were observed. On the bottom, we contrast with the approved drug, osimertinib, which was successful in both safety and efficacy. So for Pepper's approach, I'm showing here, and I'll kind of orient the figure itself in a moment, but the high level kind of takeaway is that we had an ability to identify approved versus unapproved drugs with very crisply different signatures that were interpretable in the context of oncology. And so rows again here are the drugs themselves, so unapproved 3 at the top, approved at the bottom. Columns in this case are kinases, and we're showing a color that represents the activity of these kinases, representative of a set of cell lines that are treated with that drug of interest. Now I've highlighted with the call-out boxes, so in the unapproved case, we saw that the drugs predominantly activated many pro-oncogenic kinases, so detrimental for treatment of the disease. In the unapproved drug scenario, we actually see, in the approved drug scenario across the bottom, we see the opposite. So we actually see a suppression of, in this case, kinases that would be pro-oncogenic. So we're actually doing better at resolving the disease, and a very clear distinction between the red increase seen in the unapproved and the blue suppression seen in the approved. So really highlighting here that in early preclinical studies, Pepper's transomics approach was able to separate out the approved versus unapproved drugs. So think of in the future applying this to a scenario when you have a set of drugs where you're making a decision of what one should we bring forward. You can make that decision from earlier studies by applying transomics to have this global functional causal understanding of the drug and drive towards actually making this decision much earlier, saving the ability or saving those costs of failures of a drug. So I'll round out here just a little bit more on the cost side to give some bigger picture context as well. So I mentioned at the onset that in the EGFR mutant lung cancer case, it was about 2,000 patients that received in clinical trials a drug that was not approved, that could have received a better treatment if a decision was made earlier on what drug should be moved forward that was successful. And secondly, again, this was about $400 million that was spent that was wasted, that was not towards an approved drug that could have been used if having earlier decision making towards something else. Now, if we bring this perspective across cancer overall on an annual basis, the estimates come into about 6,500 patients per year receive a drug in clinical trials that at the end of the day was not approved. And the cost of this is on the order of about $650 million annually. So there's a significant patient and cost benefit towards making these decisions much earlier on what drug is most likely to be successful. And so to bring this technology forward, to bring this to patients, to grow Pepper, just really what are we looking for and what are we excited for the community here to help support? So the first is highlighted at the start. We do work with pharma on programs as well as our own drug programs, but we do partner with pharma to answer critical questions that they have for their own programs. So if you're at pharma or know someone who's looking for a technology to help to answer these challenging questions around what is happening in disease or what is the drug doing, would be happy to connect and discuss more what you're looking to do. Second is just looking to connect with investors to help support building out our own platform and internal drug pipeline as well. And lastly, as we grow the team, always excited to connect with passionate folks to look at growing our internal expertise to truly reach our goal of ending untreatable diseases. So to round out a little bit more on the future that we see for patients and for what, when 5-10 years down the line, this is used across the industry, what are we really seeing and building towards? And so if we think about the trajectory of how drug discovery has gone, We started, say, in oncology in the past with a very coarse definition of what disease is. We started by saying a cancer is defined by the tissue of origin. It had some benefit for patients, but predominantly was, again, lacking in its ability to truly treat a successful population of patients because of that coarse definition of what cancer is. Today, we've done better in the industry by bringing in definition of genomics and mutations to say, okay, we need to better refine by tissue and mutation type what cancer is and what the disease is that we're identifying a treatment for. So growing to helping patients further, but there's still a large gap in those patients that can actually respond to and over time receive a successful treatment. So what Pepper is building towards is a future where we then we define disease by a transomic signature. So further refining this fingerprint of disease to include information that's global, functional, and really can understand what is the most accurate definition of a disease that we can find a treatment for that a patient will respond to. And at the end of the day, they will go back home to living their life. And that's why we're here, and what at Pepper our whole team is waking up every day to get closer to building, is ending untreatable diseases so that we can all go back to living our life. And with that, I'll close for the slides. Would love to take if there's any questions that you have, Dr. Gershon, or the audience members have as well. So
Absolutely, breathtaking technology, and now you have traction with industry leaders, and it's all about putting all of this into an action-driven platform. So I am so impressed by the breadth of problems that you are tackling. And then, again, to reiterate what we talked about in the beginning of the conversation and the thematic platform on which the BetterHealth webinar series is resting, which is the notion of complexity in problem solving requires collaboration partnerships. And so, with that, I would say my, last question would really be looking at the future. I think appropriately, you've highlighted the three, what I would call short and maybe even intermediate focuses of the company. But let's take a broader leap, 3 to 5 years out. I mean, this is really part of an overall pivot, in how we're looking at disease. And I think in your last point was really outstanding in terms of its vision. Typing, cancers within a certain, channel, somewhat arbitrary, but and somewhat artificial. And most importantly, even if you don't agree with those two statements, not complete.
Yes.
And so what I am so impressed with here is the completeness of the vision and the ability to tackle this. I mean, this is not a short-term issue here at all. So I want to take a look at, let's say, a 3 to 5 year crystal ball. Where does your incredible vision see the points of collaboration in a first kind of corridor? So what are the industries that you see participating in this? You talked about a little bit about in silico and how you could be additive in terms of value there. I certainly put a check in that checkbox. So industry collaboration, where do you see that in the next 3 to 5 years? And then the second is, how do you see this in terms of the impact on the patient? We talk about speed and healthcare economics and, the ability to drive things faster and faster. But what does that ultimately mean, for the patient? So those industry and patient channels, I'd love to hear your thoughts on that.
Yeah, 100%. So first, just building on the industry side. So, I mean, we have worked with and see building a repertoire of folks on the pharma, on the biotech side of folks that are bringing forward their own pipeline programs and bringing forward assets. This could be folks starting early on at the target discovery stage saying, hey, I want to go after this area. Help me to see what's the right direction to go after. Can also be folks who have assets that are looking to better understand which one to move forward into the clinic, which, even could be an indication expansion type question, could be a biomarker question. So essentially, on either side of having a molecule, either it's getting to that point or after on the characterization half, that's where Pepper shines. So we talked about in silico of their expertise of designing the molecule itself, so we can help to answer what to design against, and then help to characterize that molecule once that compound exists. And so it's really, we fit in and plug into across the drug discovery pipeline, and are thrilled to help, again, at the end of the day, bring our insights to patients through other partnerships that we establish as well. I'll pause there before going to the patient half to make sure I've routed out on your question.
No, that's perfect. I mean, what I would say is intersecting between the industry, and the patient is definitely a shout out, to some of your supporters here. Mm-hmm. And so I would be remiss if I didn't make mention of the Merck Digital Sciences Studio. I think they're doing an absolutely fantastic job. People like Jeff Hannigan are really leading the charge in supporting companies like yours. And at Endeavor, we are looking for collaboration channels within Merck and within the Merck affiliates, including their venture capital group. So I really want to applaud them. You're part of an incredible portfolio. And so I don't have any more questions about industry except to applaud industry for recognizing great women leaders, scientists, entrepreneurs like yourself, great business leaders within the emerging healthcare community. And then love to hear your thoughts about kind of on the patient side, which is not so much concerned about the processes, in the financing of all this, but on a very personal side.
Yeah. So first, I will note the similar shout out to the MDSS program and Jeff and all the folks, Hamdi and Alexa behind that. Really great vision of what they're bringing forward, a fantastic cohort that we've been a part of, and would encourage you're in an early stage company in the digital health and biotech space to apply. We've really appreciated the support that they provided on multiple fronts. So yes, completely echo.
Great team there.
Yes, the patient side then. So what we often think of on the patient side is, as your diagnosis of the disease is at that trajectory, starting from that doctor's appointment, what happens, right? Today, oftentimes, the patient, and I can relate to this with my family as well, you can go to the doctor's office, diagnosed with a disease, and sometimes there's a trajectory of there's no treatment available where they can monitor, they can measure, but there is no option. Sometimes there's a scenario today where you, say in oncology, receive a treatment, but you respond for a short amount of time and then the disease resurges. And so that is where today we say we are still failing patients in both of those scenarios, because there is no ability for that patient and their experience to go back home and continue living the life they had before that diagnosis. And so the change that we see at Pepper bringing stems from that first appointment. So when you go to the doctor, you're diagnosed. Step one, there is a treatment for your disease. So there is an option, a therapeutic that will resolve the disease, and you will continue to respond to that treatment. And that ability to continue to respond, to have the disease symptoms and the disease itself gone, is what we're pushing towards, where you can, at the end of the day, have a treatment you respond to and go back home to living your life. And that's what my family didn't have for my father, and what John's family didn't have for his grandmother. And that's why we're here to change that. So at the end of the day, you can go back to doing what you want to do with your family, go sailing in New York and enjoy. So that's why we're here.
Fantastic. I want to again thank everyone who tuned in today. There's more people who have signed in. Kamaswara has made some great comments and always contributed He's signing in from India. We have Aurel and Gabriel. I want to thank you so much for joining. And then the other people who I mentioned earlier, including Dr. Burla, who I believe is going to be speaking with us later this month about some amazing things happening in her company. So thank you all for your support, your contribution, and thank you, Dr. Strasser, for coming on today to really give us the wow of what your company is doing, both on a very personal level. Thank you for sharing those personal stories, and on a science side, and crystal balling things for a look into the future where we can expect amazing, greater predictive value in terms of bringing drugs to market, probably a greater sense of design of these drugs, and certainly a real understanding about the ability to make cost-effective early decisions in this process, which allows money to be effectively used. And that is a whole other area where your contribution, I think, has a long leg and long impact to it. So thank you for all of the things that you've brought to the awareness of the community today.
Thank you very much. And to everyone for joining, and thanks for a great discussion, Dr. Gershon.
Fantastic. Stay on for just a minute, and I'm just going to sign us off here. See you all next Tuesday when we will be back on our regular scheduled time.